In this video, we're going to cover glomerular filtration, which is the first step in urine formation. This is beautiful, so let's start this lecture by taking a look at the overview structure of a nephron. Nephrons are the functional units of the kidney, and they consist of two parts. The first part is the renal corpuscle, which consists of the glomerulus, this ball of capillaries that is surrounded by Bowman's capsule. Blood flows into the glomerulus through the afferent arteriole. Think afferent, blood has arrived at the glomerulus, which is this ball of capillaries, and it exits via the efferent arteriole. Efferent equals exit. Blood leaves the glomerulus through the efferent arterioles. The glomerulus is the filtering structure. Plasma gets filtered through the glomerulus and forms the glomerular filtrate, or simply just filtrate. This filtrate then leaves the corpuscle and passes through the rest of the nephron. It enters the tubules where the processes of reabsorption and secretion happen. And finally, excretion. It's converted into urine. So the second part of the nephron are the renal tubules. We have the proximal convoluted tubule, which is this part here that connects with Bowman's capsule, the loop of Henle. Loop of Henle is short for cortical nephrons, whereas for juxtamedullary nephrons, the loop of Henle goes all the way down to the medulla. Then we have the distal convoluted tubule, and finally the collecting duct. So the amount of any substance excreted in the urine is equal to the amount filtered plus the amount secreted minus the amount reabsorbed. All right? That's the overview process. Let's now subtract complexity and break down the structure of the renal corpuscle and take a closer look at the filtration barrier. Okay, like we mentioned before, look at this masterpiece there. The renal corpuscle consists of the glomerulus, which divides into several branches, and then we have a fluid-filled capsule called the Bowman's capsule, which connects with the proximal tubule. The fluid that is filtered from the glomerulus into Bowman's capsule, or space, must first pass through a three-layered glomerular filtration barrier. There are three layers that make up the glomerular membrane. We have the glomerular capillary wall, or the fenestrated endothelial cells, the glomerular basement membrane, and the inner layer of Bowman's capsule, which are the podocytes. The way we're going to break this down is we're going to go through the glomerulus first and take a look at the first two barriers, and then we'll take a look at Bowman's capsule and then put everything together. It's going to be a walk in the park, okay? Let's take a look at the glomerulus first. Let's take a look at the overview picture, and then we'll zoom in. The glomerular capillaries are lined by endothelial cells, specifically fenestrated endothelium. It's supplied by the afferent arteriole and drained by the efferent arteriole. So there's the glomerulus, and there's the endothelial cells, and blood flows through the afferent arteriole, okay, which then circulates through the glomeruli and leaves the efferent arteriole. Let's zoom in closer. We have the capillary lumen at the top and urinary space at the bottom. The glomerular capillary wall consists of a single layer of endothelial cells. Like all blood vessels, it's internally lined within endothelial cells. So these right here are flattened endothelial cells. They are closely joined together with pores between them that form the capillary walls. They also have these large openings or holes known as fenestrations. They are about 50 to 100 nanometers in diameter. Okay, here's another drawing showing this. We have the endothelial cells, and these dark holes are the openings, the fenestrations. This drawing kind of looks like a burrito, okay? But that's what a fenestrated capillary is. It has these holes or openings. The word fenestra means window, so these holes are like windows. And these openings allow for the rapid movement of fluid, water, and solutes across the capillaries, while also preventing cells from entering. This is what's going to help with filtering plasma, okay? We're going to go through what can and can't pass through this barrier in a minute when we get to the selectivity of this filtration barrier. That's the first layer, the fenestrated endothelial cells, with the openings having a diameter of 50 to 100 nanometers. The next layer surrounding the endothelium is the glomerular basement membrane, and this membrane has 3 sublayers. This is what separates the vasculature from the urinary space and is formed by the glomerular endothelial cells and podocytes. On the endothelial side, next to the endothelial cells, is the lamina rara interna. Then we have the central lamina densa, this thick middle layer, and then the lamina rara externa on the epithelial side next to the podocyte layer. Both the lamina rara interna and externa are composed of proteoglycans, heparin sulfate proteoglycans, whereas with the lamina densa, it's mainly composed of type 4 collagen and laminin. Collagen helps with the structure and maintenance of this basement membrane, okay? Here's the thing though. The heparin sulfate proteoglycan is negatively charged, which is important in restricting the movement of negatively charged molecules across the basement membrane, which we'll go through. Just know that the basement membrane is negatively charged, mainly due to the heparin sulfate. So these right here, negatively charged. Okay. That's the endothelial cell lining and the glomerular basement membrane, which essentially make up the glomerulus and the first 2 layers of the filtration barrier. Let's go back to the overview picture and move on to Bowman's capsule and Bowman's space, and then break down the 3rd layer of the filtration barrier. Bowman's capsule is made up of simple squamous epithelium. And then we have Bowman's space, or urinary space, because it's filled with filtrate. With that said, there are 2 poles in the renal corpuscle. We have the vasculature pole, this is where blood enters and exits the glomerulus, and the urinary pole, where filtrate leaves Bowman's capsule and enters the proximal convoluted tubule. So Bowman's capsule is made up of the parietal layer, the blue cells here, and the visceral layer, which are the podocytes. The podocytes line the outer surface of the glomerulus and make up the inner layer of Bowman's capsule, this nice green layer here. This is the outermost portion of the filtration barrier, which covers the basement membrane and glomerular capillaries. And these podocytes, the word podo means foot, so they have foot-like processes called pedicels that surround the outer surface of the capillaries. Again, we have the capillary and then the basement membrane, and the podocytes surround the outer surface of the capillaries. Let's zoom in. The foot processes have these gaps called slit pores or filtration slits, through which the filtrate moves from the capillaries to the lumen of Bowman's capsule. So they wrap around the basement like an octopus, with the gaps between called filtration slits. These slits are about 20 to 30 nanometers in diameter. Now, between these filtration slits, or podocytes, are these transmembrane proteins called Nephron. This is a thin diaphragm between the filtration slits that act as a final filtration barrier that only allows substances that are less than 8 nanometers in diameter to pass through. This is called the slit diaphragm, and it's made up of nephron, which is also negatively charged. So nephron repels negatively charged proteins like albumin. All right? That's the Bowman's capsule. Let's now put everything together and summarize the components of the filtration barrier, or the glomerular filtration apparatus, and go through what can and can't pass through this barrier into Bowman's space. So there are 3 parts. The first layer is the fenestrated glomerular capillary, then the glomerular basement membrane, and finally the podocytes, the foot processes with the filtration slits. So if a substance is between 50 to 100 nanometers in diameter, it can pass through the capillary pores or fenestrations. And then if it's not repelled by the negatively charged basement membrane, it will go through and pass through the filtration slits between the foot processes, if its diameter is less than 8 nanometers, and into Bowman's space, where it's now called filtrate. And from here, the filtrate moves to the lumen of the proximal convoluted tubule. Those are the 3 components of the filtration barrier. Now, the selectivity of the barrier is really important for renal function. It's based on both molecular size or weight and electrical charge. Low molecular weight substances can pass through. This includes water, electrolytes and small ions such as sodium, chloride, potassium, calcium, magnesium, and bicarbonate. What else can pass through? We have glucose, amino acids, vitamins, urea, uric acid, creatinine, and fatty acids. Now, high molecular weight substances, on the other hand, are restricted. The amount filtered becomes smaller as the molecule becomes larger. So what can pass through are blood cells, plasma proteins, large anions, and protein-bound minerals, and also serum albumin because of its negative charge. Albumin is actually in the molecular weight range, but it still can't pass through because of its negative charge. So the second factor is the electrical charge. Like we mentioned before, the filtration barrier contains negatively charged glycoproteins, the heparan sulfate proteoglycan, or the heparan sulfate membrane. Therefore, negatively charged molecules are repelled. They can't pass through. All three layers of this barrier are negatively charged. Even if a substance is small enough, if it's negatively charged, it won't be able to pass through. An example is albumin. Recall, opposites attract and like charges repel. So the negatively charged barrier is like, sorry mate, you're negative, I can't let you pass. So the filtrate is normally free of plasma proteins, and positively charged macromolecules are filtered more and negatively charged molecules less for any given size compared to neutral molecules. And this is because of the negatively charged barrier. And one more thing, only macromolecules are restricted by the negative charges in the filtering barrier. Both mineral anions and low molecular weight organic anions are unaffected. So ions like chloride and bicarbonate are freely filtered. They can pass through even though they have a negative charge. Okay, let's go back to the overview diagram and continue. We mentioned that the filtrate is normally free of proteins, but sometimes some macromolecules, proteins such as albumin, get passed and they get stuck in the glomerular basement membrane. And this is where the mesangial cells, these majestic cells, come in. And what these mesangial cells do, okay, these right here, they are mesangial cells. What they do is they phagocytose any molecules or trap residues from the mesangial space to keep the filtrate free from any plasma proteins. Because remember, we don't want any proteins, negatively charged macromolecules within this space. So they phagocytose any trap residues, okay? Another cool function of these majestic cells, these mesangial cells, is that they have contractile properties. They're modified smooth muscles that are able to contract and relax to regulate capillary flow, which is the amount of blood that's coming through the afferent arteriole. That's the function of the mesangial cells. Before we move on to the glomerular filtration rate, let's go through the Juxtaglomerular Apparatus, which is the structure next to the glomerulus that's really important in regulating kidney function, such as maintaining blood pressure. The word juxta means next to. Don't confuse this with the juxtamedullary nephrons. The Juxtaglomerular Apparatus contains three types of cells, which are located at the vascular pole of the renal corpuscle. These are the juxtaglomerular cells, also known as JG cells or granular cells, macula densa, and the mesangial cells, also known as the extraglomerular mesangial cells. Let's go through the JG cells or granular cells. JG cells are specialized smooth muscle cells from the afferent arteriole that produce, store, and secrete renin, which is part of the renin-angiotensin-aldosterone system or RAS pathway. Renin is important in maintaining blood pressure. JG cells secrete renin when the smooth muscle detects a decrease in renal perfusion pressure, or when macula densa cells sense a decrease in sodium concentration in the tubular fluid. The second cell type is the macula densa, which are the cells in the distal convoluted tubules located in the region where the afferent arterioles enter the glomerulus. It's this tubule here that's close to the arterioles of the glomerulus. These cells monitor sodium chloride concentration in the filtrate. Then we have the mesangial cells, which we covered, and these cells are said to also communicate with JG cells, the granular cells, to produce renin. But that's the Juxtaglomerular Apparatus. Let's move on to the next part of this lecture and break down the forces involved in glomerular filtration, because in order for filtration to occur, there must be some sort of force to drive or push the plasma to move fluid in the glomerulus across the glomerular membrane. Let's label the afferent and efferent arteriole first, all right? So this is the afferent arteriole, and we have the efferent arteriole. Now, the volume or amount of fluid that is filtered from the glomerulus into Bowman space per unit time is known as the glomerular filtration rate, or the GFR. This is the rate at which substances get filtered through the glomerulus every minute. On average, it's 125 milliliters per minute. Now, you're probably thinking, did you pull that number out of thin air? Where did that come from? So let's slow down for a second and break down where we got the 125 milliliters. The kidneys receive about 20% of the cardiac output. It enters through the afferent arteriole and leaves through the efferent arteriole. And this is about 1 to 1.2 liters or 1,200 milliliters per minute. And out of that 1.2 liters of renal blood flow, only approximately 625 milliliters per minute is used in this filtration process. Now, here's the thing. Out of the 625 milliliters per minute, only 20% of the plasma that enters the glomerulus is filtered, which is the fluid in Bowman space. So GFR, the glomerular filtration rate, is approximately 125 milliliters per minute, or 180 liters per day. This is also referred to as the filtration fraction. Okay, so about 20% of the plasma flowing through the kidney is filtered. What a genius beast. Absolutely beautiful. Now that we know the GFR, let's go through how we can determine the GFR and the factors that influence the glomerular filtration rate. We can determine the GFR by the net filtration pressure and the permeability and surface area available for filtration. These characteristics, I'm going to say, of the glomerular membrane, surface area, and permeability are referred to as the filtration coefficient. Okay, as a result, GFR equals KF, filtration coefficient, times the net filtration pressure. Let's break this down further. Let's take a walk in the park and go through net filtration pressure. All right, remember what we said before? We need some sort of force to push the plasma, okay, to move fluid through the glomerulus. So the NFP, the net filtration pressure, is the sum of the pressures across the glomerular membrane. There are 4 pressures we're going to look at, and these are known as starling forces. These include the hydrostatic pressures and the oncotic or colloid osmotic pressures that are occurring on both sides of the capillary wall. Let's go through the first pressure, which is the glomerular capillary hydrostatic pressure, PG. It's the fluid pressure that's trying to push fluid or plasma out of the capillary and into Bowman space. So it favors filtration. And the average glomerular hydrostatic pressure is 55 millimeters of mercury. Okay? That's one force. The next hydrostatic pressure is the one in Bowman's capsule, PB. It's called Bowman's capsule hydrostatic pressure, and this opposes filtration. This is the pressure that's built up in the capsule, the pressure exerted by the fluid, and it wants to push fluid out of Bowman's capsule and back into the glomerular capillary. So those are the hydrostatic pressures across the capillary wall. The colloid osmotic or oncotic pressure is from the protein concentration differences across the capillary that create an osmotic force. So recall that plasma proteins can't be filtered. So they are in the capillaries, in the glomerular capillaries. So there's a difference in protein concentration and water concentration because the concentration of water is technically higher in Bowman's capsule than in the glomerular capillary. So it's going to create an osmotic force that favors fluid movement or water movement from Bowman space into the glomerular capillaries. So colloid osmotic pressure is the pressure that's exerted by proteins that are trying to pull fluid or water towards them, such as albumin. It tries to keep the fluid within the capillary. It's like, hey, no, you can't leave, okay? In the glomerulus, this is pi g, and on average, it's about 30 millimeters of mercury. This pressure opposes filtration. It wants to keep the fluid, the water, within the glomerular capillaries. It doesn't want it to leave. It's pulling fluid towards them. In Bowman's space, this is pi b. Here's the thing though. Remember what we said earlier, that there's usually no protein in Bowman's capsule. There's no protein in the filtrate. So essentially the colloid osmotic pressure of the Bowman's capsule fluid is considered 0, and we don't really include it in the overall equation. All right, so this whole thing that we've just covered is called the net filtration pressure. The forces that are favoring or encouraging filtration are the glomerular hydrostatic pressure and Bowman's capsule colloid osmotic pressure. And the forces that are against filtration are the glomerular capillary colloid osmotic pressure and Bowman's capsule hydrostatic pressure. And if we calculate this, the net filtration pressure is 10 millimeters of mercury, which is the net difference that encourages filtration. Okay? Now, the actual rate of filtration, or glomerular filtration, also depends on the filtration coefficient, the surface area and the permeability of the glomerulus. The smaller the surface area, the smaller the GFR. And if we had a bigger surface area, the greater the GFR, because there's more area for filtration. Cool. Pretty straightforward, right? Now, when it comes to permeability, it's about how many openings there are, or how many holes. So the more openings, the higher the GFR for fluid to pass through. So overall, the GFR is influenced by both increased and decreased filtration coefficient, as well as any fluctuations in NFP, net filtration pressure. And this leads us to what happens to the GFR if there are any changes that occur to either the hydrostatic pressures or colloid osmotic pressures, because GFR is not a constant value and is controlled by physiological processes, okay? Starting with the glomerular capillary hydrostatic pressure, GHP, GFR increases when glomerular hydrostatic pressure rises, while GFR decreases as glomerular hydrostatic pressure decreases. There are 3 factors that influence glomerular hydrostatic pressure: arterial pressure, afferent arteriolar resistance, and efferent arteriolar resistance. If your blood pressure increases, this will increase GHP. And if your blood pressure decreases, this will decrease GHP. Now, if we take a look at the constriction or dilation of afferent or efferent arterioles, if we constrict the afferent arteriole, this will decrease GHP and decrease the GFR. If we dilate the afferent arteriole, this will increase GHP and GFR. On the other hand, if we constrict the efferent arterioles, this will increase GHP because there would be resistance to outflow from the glomerular capillaries, but only if it doesn't reduce renal blood flow too much. And if we dilate the efferent arteriole, this will increase GHP, thus increasing GFR. Now, with the hydrostatic pressure in Bowman's capsule, normally changes in Bowman's capsule pressure don't act as the principal mechanism that regulates GFR, but let's say there's an obstruction anywhere along the tubule. This will increase the tubular pressure all the way back to Bowman's capsule, which will increase the hydrostatic pressure and thus decrease GFR. So generally, increasing the hydrostatic pressure in Bowman's capsule reduces GFR, whereas decreasing the pressure raises GFR, okay? And finally, with the glomerular capillary colloid osmotic pressure, recall that the amount of plasma that is filtered is expressed as the filtration fraction, which is about 20%. That is, about 20% of plasma entering the kidney is filtered into Bowman's space. So if you have low proteins in the blood, for example in liver disease, this will decrease your colloid osmotic pressure, which tends to increase GFR because you're going to be losing fluid more than usual into Bowman's space. On the other hand, if you have a high plasma protein concentration, what's going to happen? More fluid is going to be retained because it's going to be pulling water towards itself, which will increase the colloid osmotic pressure. So An increase in pi g is directly proportional to the filtration fraction, which is the amount of fluid that is filtered. And if the filtration fraction has changed, there has likely been a proportional change in pi g as well, which has affected GFR. Thank you for watching this video. Make sure you subscribe to EKG Science so you don't miss a single lecture. And remember, subtract complexity and slow down. To study the next lecture, simply click the next video or you can view the entire playlist. Hey, stop procrastinating!